good afternoon, or good morning, depending where you are located. Thank you very much for sharing your time with us today in this live webinar, where we will be talking about the screening for chemicals with thyroid hormone disrupting effects using Zebrafish Embryo. Based on our recently published paper from Elsevier in Reproductive Toxicology. So at the end of the webinar, you will have a slide with a QR and a link where you can download the paper if you haven't received or downloaded it yet.
Oh. Yeah.
So today we will be presenting this recently published paper based on the work for the validation of the thyroid inhibition assay in zebrafish done at BioBite. And I will be presenting with Andrea Weiner, Serial Study Director and Lab Manager at BioBite. She's Bachelor of Biotechnology, Developmental Biology and Embryology in Biotechnology Entrepreneurship, and PhD in Biological Sciences and Molecular Biology at the University of Rosario in Argentina, with more than 20 years of experience working with the zebrafish animal model, especially in developmental biology, teratogenicity, and disease model generation. She has extensive zebrafish expertise in different research centers worldwide, especially in the ones that she stay in Norway, England, Chile, or Spain. And for those that don't know me, I'm Aranza Muriana, an MBA and B.Pharm in pharmacy and MCS in research development and innovation of new drugs from the University of Navarra. And I've been trained in zebrafish at the beginning in the Salk Institute in California and US, and then in Spain with more than 18 years of expertise in this animal model, especially in toxicology, safety, and in some pharmacological areas such as neurobiology. So our talk will be divided in different areas. We will start with an introduction on endocrine disruptors. Then we will explain a bit on thyroid disruption in early drug and chemical development and the regulation and guidelines that has been developed lately in this area. Then Andrea will explain about our thyroid disrupting screening in the zebrafish with our research and experimental design and the results we got in the validation and the conclusions. And finally, we will specifically remark the zebrafish thyroid inhibition assay advantages, some BioBite general characteristics and information on other assays that could complement this one. And then you will have to do time to questions and answers. I will ask everyone to please mute your microphones, please, while we are doing the. So we would like to define endocrine disruptors, and we want to do it based on the US Environmental Protection Agency definition that says that they are agents that interfere with the synthesis, secretion, transport, binding, or elimination of natural hormones in the body that are responsible for the maintenance of homeostasis, reproduction, development, and or behavior. They can be classified in different ways. Diamantic and Darasis et al. Classify them in two categories: natural chemicals found in the human and animal body, like phytoestrogens, or synthesized chemicals, especially synthetic chemicals used in industrial solvents or lubricants and their bioproducts, plastics, plasticizers, pesticides, fungicides, or even pharmaceutical agents like the estevestrol, for example. But if we go to other kind of classifications, we could also go to the characterization done in three groups by the occurrence, according to Gore et al. From 2040. Like pesticides, that they can be highly sensitive toward the reproductive and neural system of the organisms, such as DDT, for example, or chlorpyrifos. Chemicals in products, that they are present in products that are used in our day-to-day life, starting from children's products, electronics, personal care products, and many of them even in direct contact with absorptions, direct ways like skin or mouth or hands, but basically classified in two subclasses: children's products and electronics, and finally food contact materials such as bisphenol A that are used frequently in plastic-based containers especially in epoxy-based line cane foods, also in direct contact with food and going through the oral dosing way. So, summarizing, we can say that endocrine disruptors are present in the air we breathe, in the water we drink, and even in the soil or our food is cultivated. And one class of endocrine disruptors, especially the neuroendocrine disruptors, are recognized as environmental pollutants that are found to mimic or categorize or modulate the synthesis and metabolism of neuropeptides, neurotransmitters, or neurohormones, which result in altered production, reproduction, development, ability to deal with challenges, and many other abnormalities. Therefore, they are known to mainly work by altering hormonal and homeostatic system of living systems involved in the regulation of several processes like metabolism, sexual development, insulin production, utilization, growth, stress response, gender behavior, reproduction, or even fetal developmental of the living body. And they are also seen in wildlife. Because the diverse mechanism of action of the ADCs in the human body, it's very difficult to establish a relationship between them. More even, sometimes the metabolites of those ADCs can be the hazardous or even more hazardous than the parent compounds themselves. So generally, it's not possible to determine whether a compound is an ADC or based on its structure. Despite from different observations, it's known that phenolic ring with no halogens, sulfonal functional groups, chlorine and bromide substitutions next to the OH group in phenolic rings can be required for ADC activities. And when was the regulation checking the ADCs? Well, in the 1990s, the public became aware of the issues regarding endocrine disruption, chemicals following reports of decreased sperm quality and increased incidence in endocrine-related cancers that linked these health concerns to human chemical exposures. But the study of the endocrine disruption began with a famous meeting on the Widespread Conference Center, Racine, in Wisconsin in 1991. So it's less than 20 years, or more than 20 years, 22. And it was organized by Theo Colford and co-workers. And actually, this meeting made the first paper in endocrine disruption from Colford and Clement, published in 1992. In 1996, an advisory group of endocrine disruptor testing and assessment, the ETA, was established at the OECD to develop new and updated existing tests guidelines in order to identify endocrine disruption chemicals. But in the 2000 year, for example, there weren't so many papers if you check them based on the endocrine disruptor concept. So in Science Citation Index, for example, in May 2002, it only gave 1,346 research articles published, being the first paper from Colbert and Clement et al., the one that we mentioned from 1993. And actually, the OECD guideline number 150 on the standardized test guidelines for evaluating chemicals for endocrine disruption series was published in 2012 in Paris. So it's less than 15 years. And the review of this OECD guideline was in 2018, back to 5 years. So it's really a very new area, endocrine disruption, and it needs a lot of review and a lot of studies and research yet. The OECD number 150 guideline organized available test methods at different 4 levels, depending on the existing data, on if they are in vitro or in vivo, and providing a general description of each standardized test guideline used for endocrine disruption screenings. And the recent review from 2018 even mentioned some validated or even invalidation process new tests, also based on new alternative methods such as Zebrafish. And this extended review from 2018 also mentioned a 5th level, including the Zebrafish. So this is why we want to show you how this OECD guideline includes different levels and somehow is trying to be modernized, review the new ongoing alternative methodologies, and update it with new assays. If we check level 2, in vitro assays providing data about selected endocrine mechanisms, pathways, in mammalian or non-mammalian, we can see that they already mentioned the thyroid disrupting assays as one of the assays to be available. And they also mentioned high throughput screens. If we could combine two, we would say that the Zebrafish thyroid disrupting assay could be a high throughput or high content screening to be included in this level 2. Why not? If we go further to level 4, in vivo assays providing some mechanism of action on endocrine disruption, we can find the reproduction and developmental toxicity screenings based on the OCD guideline 421 and the developmental neurotoxicity studies based on OCD guideline 426. Both can be indirectly affected by the endocrine disruption, and especially the thyroid disruption. So that's why we feel that it's very interesting to combine endocrine disruption assays in zebrafish with other developmental and reproductive toxicity screenings or developmental neurotoxicity screenings in zebrafish. And if we analyze the last level, level 5, in vitro assays, in vivo assays providing more complementary data on adverse effects on endocrine relevant endpoints over more extensive part of the life cycle of the organism, we find finally a zebrafish assay mentioned. The Zebrafish Extended One Generation Reproduction Test, a draft OCD guideline in 2018. But apart from that, other zebrafish assays could be included in those levels, as we mentioned, if we combine different assays mentioned in those tables. And that's our purpose, to validate the thyroid inhibition assay in zebrafish that can be done in a high content screening manner to detect thyroid inhibitors in tier 1. So now I leave the stage to Andrea to explain you our validation for endocrine disruption screenings in zebrafish.
So as Arantxa said, it is known that there is an increasing concern worldwide about the environmental impact of chemicals, especially the ones that can affect the endocrine system, because they also can disrupt the development, the reproductive, the neurological, and the immune functions, both in human and wildlife. Between the main endocrine pathways, we can find the estrogen, the androgen, the thyroid, and the steroidogenesis pathways. We will focus on this work in the thyroid pathway. So our research is focused on the productivity of the zebrafish for thyroid disrupting effects of new chemicals to complement other screenings such as developmental toxicity or developmental neurotoxicity assays. So we can consider that there is an increasing respecting the zebrafish as a model for endocrine disruption because it is highly popular in toxicology. It provides an ethical small-scale analysis system with the complexity of a complete organism. It enables continuous developmental monitoring. It is widely used for answering relevant issues on mammalian developmental hazards. It's also considered for endocrine regulation because the hypothalamus hypophysis is highly conserved in humans and this model, and the exposure to EDCs cause developmental malformations and reproductive dysfunctions. So we take advantage of the transgenic zebrafish line that expresses the M-cherry reporter under the control of the reporter for the hormone precursor thyroglobulin. So we all know that EDCs act at different levels of the thyroid pathway. So by revealing this terminal downstream event, any disruption of the thyroid system will be highlighted by changing the levels of the fluorescence in the reporter. So this line provides a specific expression, high sensitivity, and a concentration dose response. Here you can see different pictures of the brain of the 3 different larvae exposed to the control vehicle in this case, and in these 2 cases exposed to potassium perchlorate at different doses. So you can see from the dorsal view how the thyroid is observed, all the expression and for the expression of the fluorescence reporter. So the work is divided in 2 parts. The first one involves the systemic toxicity of the reference compounds in which by developing a maximum tolerated concentration assay, we can establish the lethality and morphological alteration of the different compounds and determine the LC50, EC50, and EC10 values in order to use these values for the second part of our work that is focused on the thyroid disruption assessment. In this case, we performed 3 different assays. One involves the use of the transgenic Zebrafish as a biosensor for detecting the EDC compounds. Then the gene expression analysis of different target genes related to specific key events of the thyroid disruption. And then to measure the T3, T4 hormone levels in order to determine the concentration of these hormones in the treated embryos by a new developed method of our group by HPLS MSMS. So we selected 7 different reference test chemicals, as you can see here in this table. The first 3 belongs to the thyroid disruptor chemicals, and they are N-phenylthiourea, potassium perchlorate, and propylthiouracil. And you can see that they act at different points in the pathway. We also select 3 different environmental pollutants like bisphenol A, benzophenone 2, and fluoroglucinol, also having different mechanisms of action. As a negative control, we use acetaminophen. For the first part, we tested the 7 compounds in our maximum tolerated concentration assay, and the experimental design consists of the exposure of the larvae from 48 hours post-fertilization to 120 hours. We treated 15 embryos per condition at 8 concentrations for each reference compound, and also we tested the vehicle control. We used 24 well plates and put 5 embryos per well. And at the end of the day, say 120 hours post-fertilization, we analyzed the embryo under the scope to detect the morphology and lethality of them. Regarding the morphology, we detected different body shape alterations, different edemas like heart edema or yolk edema, and also necrotic tissues. With all this information, we could establish the EC50, EC10, and LC50 values for each compound. After performing the assays, we could determine the values for all the compounds except for potassium perchlorate and for fluoroglucinol, because the embryos didn't show any toxicity when incubated with these reference compounds. So the following thyroid disrupting screening were carried out at concentrations below the EC10 values to avoid any interference of systemic toxicity. Then for the thyroid disruption screening, we designed a similar experiment where the embryos were exposed from 48 hours to 120 hours post-fertilization with 15 embryos per condition. And in this case, we used only 5 concentrations per compound plus the vehicle control. We also tested the embryos in 24 well plates with 5 embryos per well. And at the end of the assay, we take images of each embryo to analyze the fluorescence of the reporter gene. With this assessment of the fluorescence, we could perform an ANOVA analysis to compare the results of the reference compounds with our vehicle control and to perform the dose response curves. Here we have the results for the reference thyroid disruptor chemicals, and you can see that for N-phenylthiourea, propylthiouracil, and potassium perchlorate, we detect significant differences, increases at the highest tested concentrations for the 3 compounds. So an induction of the thyroglobulin was detected. And for the negative control, we didn't observe induction of the fluorescence in these embryos. So we could say that no thyroid metabolism alteration was observed in the embryos exposed to acetaminophen. For the environmental pollutants compounds, we detected an induction in the fluorescence, and it was significant for benzophenone 2 and fluoroglucinol. However, for bisphenol A, we couldn't detect an induction in the fluorescence at any of the concentration tested. So no thyroid metabolism alteration was observed for this chemical, and we could classify until now, for now, as a false negative. But as a summary of this assay, we can say that the sensitivity of the fluorescent assay allows us to detect the thyroid disruptor effect at environmental relevant concentrations for almost all the compounds. Regarding the gene expression analysis, we detected the 3 different target genes, TSH beta, TG, and TPO, and also at 5 concentrations per compound. We performed RT-qPCR, and here you can see the results for the reference thyroid disruptor chemicals, and for propyltiouracil, potassium perchlorate, and phenylthiourea. We observe an increase in the expression of the 3 target genes, highlighting the effects of these chemicals in the thyroid pathway. For the negative control, the acetaminophen, for TG and TPO, we didn't observe any change in the gene expression. However, for this TSH beta, we observed a slight increase in only 1 concentration by the RT-qPCR. But we will say that this is now a dose response, and the other 2 genes are not modified their expression. So we classify it as our negative control. And for the environmental pollutants chemicals, we also observe a significant increase for benzophenone 2 and fluoroglucinol at the highest tested concentrations. But for bisphenol A, we didn't observe any change in the concentration, in the expression of the target genes at any of the concentrations tested. So we also classify them with this assay as a false negative. As a summary of the results of the fluorescence assay and the gene expression, we can say that we have the fluorescence assay is a dose response induction that is the strong gene expression induction after the exposure to the reference compound was observed for all the compounds except for bisphenol A. The induction of 1 or more thyroid-related genes at the tested concentrations was also observed, and no response was detected for our negative control, the acetaminophen. So we can partially classify them as true positive. Our 5 compounds is as a false negative, the bisphenol A. And then we wanted to add more data to these 2 assays by quantifying the T3 and T4 hormone levels by a newly developed method of our group, using liquid chromatography and gas chromatography with mass spectrometry detection. In this case, we expose the embryos at the same timeline, but we expose a bigger amount of embryo, 150 embryos per condition. We only tested 1 concentration per compound, and also we detected the hormone levels in the vehicle control to compare with. And here you can see the graph for T3 hormone levels and for T4 hormone levels, where potassium perchlorate show an increase in T3 and a decrease in T4. That is quite related and is good, this relation. But we also detected a decrease for T4 levels for benzophenone 2 propyl, tiuracil, and fluoroglucinol. But fluoroglucinol show an increase in the T4 levels. So with this assay, we could study that changes in T4 levels were detected only for bisphenols 2, potassium perchlorate, propyltiuracil, and fluoroglucinol, and no effects could be detected on the T3 levels except for potassium perchlorate. So our conclusions are...
Yeah, based on what just Andrea explained about our results, we can say that the thyroid inhibition assay with the transgenic zebrafish thyroid model, complemented by the RT-PCR gene expression analysis of the related genes to the thyroid inhibition, enables a time and cost-effective assessment of the potential endocrine disruption effect of chemicals, and it can be even complemented with the T3, T4 levels if we are interested to have more information on them. The sensitivity of these assays allows to detect the effect of endocrine disruptors, especially the ones on the thyroid inhibition axis, at environmentally relevant concentrations. That way we tested the EDC-10s based on the first phase for maximum tolerated concentration to detect this EC10. And therefore, zebrafish transgenic models such as the one we use for the thyroid inhibition, but also others such as the one that is already validated by the OECD 250 guideline for transgenic pathways, represent a promising new alternative methodology for inclusion as an intermediate step between cell-based evaluations and conventional animal testing in tier 1 screening batteries for chemicals that impair thyroid hormone synthesis or other endocrine disruption synthesis impairments. And here we have the table of the reference compound that we tested for the fluorescent assay, for the qPCR, and how we complemented with the T3, T4. Despite sometimes we have some false negatives, like with the bisphenol A, that can be related to indirect mechanism of action or not clear mechanism of action. And that's why we think that the thyroid inhibition assay in zebrafish could be complemented, for example, with other endocrine disruption assays in zebrafish too, like the estrogenic pathway assay with another transgenic fish. So the sensitivity of this thyroid inhibition fluorescent assay, together with the gene expression analysis, detect the effect of the ADC's environmentally relevant concentration and can be confirmed at a functional level by the determination of thyroid hormone levels by LCMCs for T3, T4. The zebrafish EDC test systems provide to be an appropriate NAM, new alternative methodology that enables a time-effective, reproducible, and sensitive assessment of potential effects of EDCs on the hypothalamic pituitary thyroid axis. Moreover, the developmental toxicity assays are confirmed to show difficulties to be used for hazard assessment of EDCs sometimes, so we think that EDC classifies as not teratogenic or not toxic, while reference EDCs show their thyroid disrupting effects already at a lower micromolar range, could be complementing those, despite we found them at not developmental toxic, or despite sometimes we found them at toxic or systemic toxic. And there is a necessity for new effective endocrine disruption evaluation methods, such as the presented today for fluorescent assay on the thyroid inhibition assay, or the one for RT-PCR, but others also, to evaluate the safety profile of chemicals and to complement current teratogenicity or neurodevelopmental toxicity assays in zebrafish and in other models, other new alternative models. So those approaches will help to identify possible endocrine disruption effects of new chemicals and drugs for a proper risk assessment. And we really are very glad to see that ODC guidelines are reviewing all the time, and they are trying to be updated and including new alternative methodologies, as they have done with a new call for evaluating phenotypic screening in fish for thyroid inhibition assays. And if you want to know more about us, BioBite is a CRO with more than 18 years of experience working in the zebrafish aquatic alternative model, offering preclinical assays to pharmaceutical, biotech, chemical, agrochemical, cosmetic, or nutraceutical companies, with the high quality level of working under good laboratory practices, and also under the 3 R's, refinement, replacement, and reducement of animals, because we try to develop and design our assays below the 6 days post-fertilization, where the zebrafish start feeding independently, and they are considered under the experimental European law animals. We have a headquarters in San Sebastian in the north of Spain, near the French border, where we do all the experiments, and we have our own facility, and we have also a headquarters business development office in US apart from the headquarters. And if you don't know about zebrafish, and you are now interested more and more in new alternative models, the high genetic homology with humans, more than 82% of the genes involved in human diseases are maintained in zebrafish, the fast development and organogenesis that allows us working with 2-3 days post-fertilization fish with most of the organs already developed, that they are considered animals after 5-6 days post-fertilization when they start feeding independently based in the European Directive from 2010, 63. The small size and transparency that allow us using automation, machine learning, artificial intelligence technologies, or the high productivity because we can have more than 200 eggs per couple of adults in a week that allows our developing high content screening or phenotypic screening assays makes them a valuable tool for cost and time efficient assays. And in our facility, we have capability to handle more than 100,000 adult fish nowadays in the best quality standards and animal welfare standards. And currently, we have more than 70 zebrafish strain lines, mutant, transgenic, or wild-type lines, including the thyroid line that we use for the thyroid inhibition assay with the M-cherry fluorescent in the thyroid area, but also the estrogenic line with fluorescent, in this case, green fluorescent in the estrogenic pathway that could complement the thyroid inhibition assay with this estrogenic pathway assay, both in zebrafish. And apart from working in the endocrine disruption area, we also have teratogenicity, neurodevelopmental toxicity, neurotoxicity, immune toxicity, hepatic toxicity, cardiac toxicity, or many other toxicity and safety assays, but also efficacy assays with different disease models. So if you want to know more, you can check our papers published recently, like the ones that you see in this slide, but also our website with many articles and the papers published lately. And now, if you have any questions, Andrea Weiner or myself, we will be happy to answer them. So you can raise your hands if you are interested and open your mic and make the question. Or write them in the chat and we will translate to everyone, yeah. I think someone is raising a hand. So if you want, Leonard, which yes, you can introduce yourself and make the question. Thank you.
Yes, thank you. So it looks like a very promising model to detect thyroid activity, but I was just wondering about the test concentration, because many are, I think, above 100 milligrams per liter, which normally you wouldn't test that high. You would stop at 100 milligram per liter for an acute test already. So I'm not sure about the sensitivity.
Well, we perform the assay under the EC10 value, so the systemic toxicity, and the unit is micromolar. So you can, if you put, you can
go to the slide.
table that in which we compare the different the sensitivity of detection of each assay and for each compound. Here you can see at which concentration this compound was positive for the fluorescence assay. So we detect that our fluorescence assay is a more sensitive one if we compare the three ones. And the T3, T4 levels are very similar, the concentrations that we reach the positive one. So yes, we know that some of them are very high, like the fluoroglucinol or the potassium perchlorate, but you can see that it's 1 millimolar for fluoroglucinol or 5 millimolar in the case of qPCR. So our assay can detect more sensitive, in a more sensible way this compound, for example.
Just to add some information, I don't know if you are used to work in zebrafish, but in this case, we are not given the drug orally or intravenously. We are putting in the media, solved in water with some DMSO. And usually, if we perform the bioavailability of the compound itself in the embryo, when we get really high concentration inside the embryo, we could say that we reach usually maximum 30% of the concentration that we put originally in the media. So thinking on those percentages, the concentration that is really inside the embryo is not really the one that you are seeing here. So this is something to take into account when you are working in zebrafish and also in vitro, for example.
I'm familiar with aquatic testing, so yeah.
Yeah.
Thanks.
Okay.
Because sometimes it's a critic that we all get when we start working with someone the first time in zebrafish. But then when we do the bioavailability analysis that we can do not only for the T3, T4, but also for the compounds themselves, we realize that the concentration, the real concentration inside is not really the one that we are putting in the media at all. Yeah. Thank you for the question. Yeah, Lisa, you can ask your question.
Okay. Hi, thanks a lot for this nice presentation, really interesting. As you know, there's many activities ongoing on that. Therefore, I have some technical questions, especially about the negative control, because we just discussed in the OECD thyroid disruptor expert group that it's very difficult to find negative controls for thyroid hormone system disruption. And I was wondering if you tested other compounds and had some false positives there, because there is a similar test using zebrafish where they have this T4 antibody staining in the follicles, and ampicillin, which is normally negative in any other assay, for example with xenopus, turned out to be a false positive. And I was wondering if you tested that or others that were false positive.
Yes, we tested ampicillin and it's also negative. Its response as acetaminophen, no effect in any of the three assays in zebrafish, so we are quite confident with that, but it's not included in this work. But we tested that control because we wanted to compare with the other models.
Okay.
Yeah, but it's true that usually when we are validating our assays, the most difficult thing is finding good negative controls or reference products. It's always the tricky part. That's why we are not confident we include only 1, 2, or 3, but sometimes there is no other solutions. So
yeah, I know the struggle, yes. And if I may ask another technical question,
Yes,
I see there's no other hands raised.
Yes,
I was wondering if I understand correctly, you measure fluorescence intensity, or do you also take into account the size or the shape of the follicles, the area?
Yes. Sorry, I didn't go in detail in that part, but yes, we tested the intensive density. So it's not only the fluorescence, but also the size of the spot that we got in the images. Yes, we have an internal software that could detect the intensity density of each picture and related with the thyroid part of the image, because sometimes you can have some external fluorescence or background in the pictures, but we delete all that and take only the ones that belongs to the thyroid gland.
Oh, okay. Yeah, that's really good, because what we observe is that, of course, also the size of the follicles change. I mean, you can see it somehow as a replacement for histopathology. Also, the shape, the morphology of the organ is important to assess, and this also has an impact on the fluorescence intensity. And this makes this assay such a good replacement for higher tier test studies.
Yeah,
Thank you very
actually, you
much.
can complement this assay also with histopathology analysis, and we do also.
Yeah,
Yeah.
it's
But
challenging.
it's for
but it's
other
possible.
years. Yeah, exactly.
Okay, thank you very much.
Any other questions? So I would like to mention also that if you have questions and you are shy to ask them or you get them later, you can write any of us, Andrea or myself. You have here your emails, our emails, so you can write if you have any doubts or questions later. Oh yeah, I can see a raised hand. Lauren Lagatic, he has the raised hand.
Yeah,
Please,
hello.
Lauren.
Hi, good to see you. I just have a question regarding the GLP. Are you under GLP or non-GLP?
So this exercise has been done under GLP environment without the GLP certification, but the good news is we are just preparing our audit for GLP. So we hope that the first quarter of 2024, we will have our ecotox assays, including the thyroid inhibition assay with the GLP certification. Yeah.
Okay, cool. Good to know. Thank you.
And there are more questions.
Yes,
Ms. Eugene, yeah, please.
Yes. So how many compounds did you screen?
So for this paper, there were 7
compounds.
1
7,
negative
okay.
and 6 positives. Yeah.
Okay, so so this is have you ever tried to use screening thousands of compounds using this technology?
Not thousands, but hundreds could be done.
Hundreds.
Yeah.
Okay. Any of these results confirmed in human cell line yet? Human cell line in the relevant cell lines, like for example, the thyroid-related cell lines yet?
In cells, you mean? Well,
You are doing toxicity study in the zebrafish, but I don't think it's really for zebrafish toxicity. We worry about it's human, right? So have you tested this or these compounds? Confirm these compounds in any human cell lines yet?
No, what we do is testing our assays. Our assays, they are always done in zebrafish or other aquatic models like algae and daphnia, but we correlate indirectly those effects with the results that we see for human safety in humans, and we can say that we have high sensitivity and specificity comparing our results in zebrafish to humans. So we are using a new alternative methodology to get results not only for environmental or ecotoxicology, but also for human safety. And actually our assays are used for human safety too. In the case of thyroid inhibition, we haven't tested thousands, but in other more complex human toxicity assays like teratogenicity or neurotoxicity, we tested a lot of compounds. And in some of them, we have reached 1,000 probably, and we compare the results with what is published for human safety, and I can say that we have a good sensitivity and specificity compared to human safety.
So did you have something like before you screen when you build this model? Could you share some of the paper that you show that this model is compatible with human thyroid toxicity?
There are papers saying that zebrafish could correlate human toxicity. We could, if you want, you can send us an email and we can share some with you.
Okay,
but
that would be great.
In our case, what we were doing is comparing our result in fish with what is expected in humans, trying to do just this thing, trying to demonstrate that zebrafish could predict somehow human safety to not only environmental toxicology.
Okay. Another question is that you mentioned about HCS in zebrafish study. Do you know any big lab that have using HCS using image of zebrafish for screening? I mean thousands of when I mentioned about screening is at least 1,000 of compounds. Did you remember any people doing that kind of screening? If yes, could you please share with some information with me?
The ones that are published has not been 1,000, they are hundreds. We have, for example, several papers published with the NIH where we have tested 115, 120 or 92 compounds for different toxicity assays, not only thyroid or endocrine. In this case, we have tested neurotoxicity, developmental toxicity, cardiotoxicity, hepatic toxicity. And you can check in our website several papers testing many compounds in different assays. The ones that we have published, other entities There are many outside, but not always 1,000. That's why we don't like calling high throughput screening. We prefer to have high content, because we don't screen the number of cells that they are tested in vitro, because the in vitro is only one cell. We are working with the whole organism alive, more complex, and we are having more valuable information than only one cell.
Okay, so that is very good point. I will send you my email asking for if you can share these two type of paper with me. That would be great.
Okay, yeah, for sure.
Thank you.
Pleasure. Thanks to you for attending. Leonard Wells is also asking again.
Yeah,
please.
I thought of another question. So I think the results showed that in all cases, except when you had a negative, that there was an increase in fluorescence. But the T4 measurement showed sometimes an increase and sometimes a decrease. So is that something you expect? Do the results match together?
Yes, because there is a tightly relationship between the T3 and T4 levels, because the more active hormone is the T3. So we observe a decrease in T4 because we assume that all T4 is compensating the T3 levels of the hormone. And the reporter gene is a transcription of the precursor of the thyroglobulin hormone. So then several changes happen, in the pathway. And the last step is the T4 to T3 compensation. So we assume that we have these low levels of T4 hormones because they are compensating this increase to keep the levels of T3 in the embryos.
Thank you.
Someone was asking something.
Yeah.
Something to take into account is also that, as we mentioned, sometimes the effect is not from the original compound, but from the metabolite. And this is also something to take into account. In other cases, what we do is doing the bioavailability of the original compound and also of the metabolite, if we know it. And we have seen that sometimes the metabolite concentration increased so much and the parent compound decreased a lot. And it has happened the same with the T3 and T4. So there is a metabolism happening in the whole organism, and the T3 and T4 conversions can be there, even regulations, and also the own T3, T4 concentrations can compensate what's happening. So if anyone has any other questions, again, you can write us to weiner@biobite.ts or moriana@biobite.ts, and we will be happy to assist you with any question, doubt, or if you have any interest in our papers. They are available in the website. Also, we have a blog with interesting articles every week, and we participate in several consortia to validate different zebrafish toxicity assays. So we invite you to check our website. And if there are not any other questions, we really thank you for attending this live webinar. We encourage you to check this QR where you can scan to download the paper. And also, I would like to mention that we will be attending the American Toxicology Association meeting in Florida in November from the 12th to the 15th. We will have a booth. We will be presenting about the consortia for developmental neurotoxicity validation in zebrafish for the OCD IATA IVIV. And we will also be present there, so you can meet us and ask questions directly to us. Both of us, Andrea and I, will be attending the Florida meeting. So thank you so much for everyone. And we also have recorded this webinar, and we will share it on the online media. So if anyone is interested in listening to it again or sharing it with anyone, you will be able to do it. So thank you so much for your time and patience, and have a great day.
Bye.
Thank you. Bye-bye.
Bye-bye.
Thank you so much.
you.